Hi, my name is Gustavo Perez, and I am in Dr. Nucleus Research Lab. And my project this summer was to further explore a chloroacidamidine warhead inhibitor. Protein arginine methyl transferases, known as PRMTs, transfers a methyl group from acetylmethionine, also known as SAM, onto the guanidinium group of arginine residues within histone proteins. There are 11 isozymes of PRMTs, which are broken down into three types based on product formation. Type 1 PRMTs create ADMA products, type 2 PRMTs create SDMA products, and type 3 PRMTs create MMA products. From these PRMTs, we only focus on PRMT1, which creates ADMA products, and PRMT5, which creates SDMA products, since when studies have shown when repressed have led to defective cell mass viability. So the importance of PRMTs is that they regulate biological processes such as gene transcription, mRNA splicing, and DNA repair. If we look on the figure on the right, we see PRMT1 and 5 methylating an arginine residue within this histone protein. So when PRMTs 1 and 5 start to overmethylate all these arginine residues, they can lead to the exposure of cancer-related genes causing cancer. So in the Nucleus Lab, what we aim to do is create novel inhibitors to prevent these enzymes from overmethylating. The differences between peptide and peptoid is where the amino acid side chain is located. In peptides, it's attached onto the alpha carbon, whereas in peptoids, it's attached onto an amide backbone. And because of this, it decreases the likelihood of proteolysis in peptoids compared to peptides. This compound here at the bottom is the novel first-generation H416 chloroacidamidine warhead inhibitor, in which reported low micromolar concentrations to reach 50% activity. In the original compound, it contains a three carbon chain, which we'll call H416 propyl. And in my project, we want to vary the carbon chain in which the chloroacidamidine warhead is attached to determine if it changes potency and selectivity. So I synthesized an H416 compound containing an ethyl carbon chain and an H416 compound containing a butyl carbon chain. Upon completion of synthesis of the H416 ethyl and butyl compounds, I went ahead and ran IC50 assays to determine potency. The data on the top left is the H416 propyl data that we obtained from Brecker et al. 2022, in which reports that to reach 50% activity, we needed 8.73 micromolars of the compound, therefore being a great inhibitor. So with the ethyl data, we saw that there was decreased potency because we needed 500 micromolars in order to reach 50% activity, therefore being a poor inhibitor. However, it still retained selectivity, as we see it didn't inhibit PRMT5. Next, we went ahead and tested it against the H416 butyl compound, in which with first PRMT1, it showed that it wasn't a great inhibitor. From the concentrations that we use, it didn't reach 50% activity, therefore being a poor inhibitor. So when we ran it with PRMT5, we found interesting results. We saw that instead of it decreasing or staying stagnant, we saw that that percent activity was increasing while increasing compound concentration. This interesting result led us to believe that the H416 butyl compound against PRMT5 is not acting as an inhibitor, but rather as a substrate due to that increased carbon chain. So we then ran a KM assay that measures the enzyme's affinity for the substrate, and which confirms that theory. So for future directions, we want to resynthesize this compound, but instead of adding that chloroacidamidine warhead group, we want to go ahead and add that guanidinium group to confirm that this amine group is being methylated. So that's the end of the presentation. I would like to say thank you to Johnson & Johnson Vision Care for sponsoring the SURP program, as well as Dr. Nuckley for his mentorship, to Dr. Causey's collaboration on this project, as well as Jordan Tucker and Molly Breakers helping this project throughout the summer. And lastly, to Dr. Lane for your mentorship and leading the SURP program this summer. Thank you.